the biochemistry or the chemistry of the lipids, we know that lipids are family of organic compounds which are not soluble in water. These are water insoluble compounds or the molecules, right? These are basically fats. Fats are basically our solid fats, simple fats or the neutral fats, or these are oil preparations. And in these fats and oil preparations, they have uh, elements of carbon, hydrogen, and oxygen in different proportions. So they are mainly composed of, these are the hydrocarbons, they are mainly composed of the carbons, hydrogens, and oxygens. What is their purpose? Why do we need these hydrocarbons in this form? They are the main storehouses of the energy. They yield energy in our body. When they oxidized, when they burn in the body, they produce energy, they produce calories, and these are the storehouses of the energy as well. So in the body, they perform mainly three important biological functions. Number one, these are the structural components. We know that the cell linings, the cell membranes, the linings of the subcellular organelles, all these are made up of the phospholipids. Phospholipids are the main ingredient of the membranes. And they, by doing this structural conformation to the cells, they do not also provide protection to the cellular structure, but they also serve as the cushioning. They also provide cushioning effects to the cell from the external sources, and within the cell they also serve as a partitioning or the compartmentalization of the cellular structures or the structures which are present within the cell. So they separate them with these membranes. Okay. Second important function of the lipid is, as I told you, that they are the major storehouses. They are the major sources of energy. We know that the 1 gram of fats, when it's oxidized, it produces around 9.1 kilocalories. So they are the storehouses of the energy. And third important function is that they are the important signaling molecules, as we have already discussed somewhere, that the cell signal pathways, the cellular signaling mechanism, second messenger system, signal transduction pathway is carried out along with these lipids molecule. Along with that, they serve some additional functions such as fat soluble vitamins. Fat soluble vitamins are also derivatives of the lipids. They are needed for the various metabolic and biochemical functions by the body. And prostaglandins and the steroid hormones. The prostaglandins are the local hormones which are produced by the cells in response to the some noxious or innoxious stimulus such as inflammation, right, such as pain. Then steroid hormones, we know that all these steroid hormones are derived from the same common nucleus of sterol as that of the cholesterol. So these are the major functions of lipids in the body. Now, what about the lipids or the fats that we consume in our diet? What we do with these? Before understanding that, we should know that most of the diet or most of the fat that we consume in our diet is in the form of triglyceride. So more than 90% of our fat content in our diet is not cholesterol. We always think about cholesterol, that we are taking high cholesterol diet. No, it's not. These are the triglyceride or the triacylglycerol. So more than 90% dietary lipid consists of the triglyceride. What we need to do? We need to digest, absorb, and transport this triglyceride or triacylglycerol to its target sites and to the sites from where we can obtain some ATPs, some energy from these triglyceride molecules. How it happens? Let's see. So 90% is triglyceride. Remaining are, which are 8 to 10% are cholesterol, cholesterol esters, phospholipids, and other free fatty acids that we take in other sources of the lipids. Now, the digestion of the lipids begins in the stomach. It's not in the salivary gland. It's not in the oral cavity. It's not in the esophagus. It begins in the stomach. But a very small part of fats is start its process of hydrolysis or the engagement of enzymes with the substrate within the oral cavity or the pathway from the mouth to the stomach, that is in the esophagus. How? Because we have an enzyme which is known as lingual lipase, and its name is suggestive of its location, that from where it is coming. Lingual, that means it's coming from tongue within the oral cavity. So lingual lipase is produced by the Abner's glands, and these Abner glands are present at the back of the tongue, on the back of the tongue. So these are the little glands which produces very little minute quantities of lingual lipase. What this lingual lipase do? This lingual lipase is not going to digest the fats or the triglycerides that we eat, but what it do? It will bind with the lipids or the fats within the oral cavity and starts the process of digestion. So these triglyceride molecules are the fatty acids of short or medium chain length, are the primary target sites of this enzyme. So that means the not the long chain fatty acids, but short chain fatty acids and medium chain fatty acids, their hydrolysis, I'm not saying digestion, their hydrolysis begins within the oral cavity or in the esophagus with the help of lingual lipase. What happens next? Okay. The same triglycerides which are engaged or occupied with the lingual lipase are degraded by an other set of lipase, which is not coming from the tongue this time, but this time it is coming from the stomach. And that lipase is gastric lipase, since it is produced in the stomach. And both of these enzymes, which is the lingual lipase and the gastric lipase, they act on the triglycerides and facilitate and starts the process of hydrolysis of triglycerides. Both of the enzymes are acid stable. But the story doesn't end here. Not the lingual lipase, not the gastric lipase are going to offer full digestion or hydrolysis of the triglycerides. For this, we need to move forward and we need to look upon another lipase which is coming from another organ. If you look at this diagram, what's shown over here are some fat droplets, as you can see. These are the fat droplets which are present in the stomach, and here we have enzyme which is the gastric lipase, and this gastric lipase binds with these fat droplets and starts the process of hydrolysis of the free fat oil droplets in the stomach, or the fat droplets in the stomach. Since it reaches to the duodenum, to the small intestine, and other set of enzyme coming from some other organ, namely the pancreas secrete some other lipases which are known as pancreatic lipase. This pancreatic lipase, which is shown over here, this pancreatic lipase binds with the lipid droplet or the fat droplet and hydrolyze it. These are the intermediate structures which I will explain you later on. What are these? These are made. These structures are known as micelles. So with the action of pancreatic lipase and bile salts, these oil droplets or fat droplets are converted into some simpler structures which are known as micelles, and these micelles are actually absorbed by the intestine. So what what we have explained, what we have understood until now, that for the digestion of the lipids or the fats, we need to short and medium chain fatty acids, lingual lipase, then same triglycerides in the stomach, gastric lipase. But when it reaches the small intestine, and other enzyme comes, which is the pancreatic lipase, which offers the final digestion of these fat molecules, and these fat molecules are converted into some simpler structures which are known as micelles, and these micelles are absorbed by the intestines. Okay, how it happens actually, how these droplets are converted into the more simpler structures. This is facilitated with the help of bile. What is bile? We know that bile is produced in the liver, in the hepatic canaliculi, and it is stored in the gallbladders. So bile is a solution which is composed of bile salts, cholesterol, some little cholesterol within it, and lecithin, and some bilirubin as well. So the composition of bile is like that, that it contains bile salts, cholesterol, lecithin, and bilirubin as well. So what this bile do with this fat droplet? This bile actually emulsifies. It offers the process of emulsification for these fat droplets. As you can see, this is a fat droplet which is present, which we consumed in our diet, and now it is present within the intestinal lumen. So what happens when it reaches to the intestine? Some bile salts or the bile molecules surrounded that fat droplet, right? In the green color, these green droplets, these green structures are actually the bile salts or the bile which surrounds the fat droplet. And as you can see, what happens? This bigger, larger, tough structure of fat or the lipid is now broken down or hydrolyzed into some smaller droplets, right? This one big fat droplet is broken down into further smaller and then into further more smaller droplets surrounded by the bile salts. So this process is known as the emulsification, which emulsification provides detergent effects. What it do, it increases the surface area for its hydrolysis and the enzymes to act over it. And what it do, it offers some hydrophilic outline so that it can easily travel and transport within the intestinal medium and absorb from the intestine. So it takes place with the help of the bile. Okay. Why bile salts need to emulsify lipids? Because it increases the surface area of the fats, and which are the hydrophobic lipid droplets, and it offers some detergent properties, and these bile salts facilitate the mixing and hydrolysis of the fats within the intestine by the intestinal movements, by the peristaltic movement of the intestine. So in doing this emulsification, mixing and peristalsis, so these bigger fat molecules are converted into more simpler and smaller fat molecules which are readily absorbed by the GI. The same thing, the same mechanism in a much simplified manner. You can see the big triglyceride droplet, bile salts come into action, converts into the more smaller fat droplets on which lipases, which are present in the intestinal lumen, the pancreatic lipases, acts on that, and these triglycerides are hydrolyzed or digested or converted into some monoglycerides or free fatty acids. So this is how these monoglycerides, we know that for the absorption, what we need to do. For the absorption purpose, we need to convert some larger or bigger polymers into the smaller monomers. Example, as you have already studied with the case of polysaccharides. Polysaccharides are converted into monosaccharides to get absorbed by the intestines. Now, these triglycerides, now which are emulsified or broken down into much smaller particles, are enzymatically degraded by the pancreatic enzymes and absorbed by the intestinal cells. Now, until now we talked about three lipases: the lingual one, the gastric one, the pancreatic one. The pancreatic lipase is the final lipase which offers the digestion and absorption of the lipid. One more enzyme which is known as the pancreatic colipase. What is the function of this pancreatic colipase? This pancreatic colipase is also secreted by the pancreas, and its job is to anchor the lipase to the lipid droplet. So that means for the pancreatic lipase to act upon the, or to bind on the lipid droplet, it has to anchor, it has to be transported from somewhere to the lipid droplet, and that anchoring job is done by the colipase. That colipase इसको पकड़ता है pancreatic lipase को और fat droplet तक लेके आता है. And we already know that these fat droplets are surrounded by what? By the bile salts. So fat droplet or the lipid surrounded by the bile salts on which lipases acts to be converted into monoglycerides. And these lipases are carried or anchored by the help of colipases. Now, what about the absorption of the fats by the intestinal mucosal cells? For its absorption, free fatty acids, free cholesterol, and monoacyl glycerols are the primary products which are produced as a result of hydrolysis of triglyceride within the intestinal lumen. And these all these are absorbed with the help of bile salts and for fat soluble vitamins in the form of micelles, so that they can be absorbed. So means the triglycerides which we consume in our diet are converted with the actions of lipases and bile salts into the monoglycerides and fatty acids, and these monoglycerides, fatty acids, bile salts makes micelles, and these micelles are absorbed by the intestinal mucosal cells. We will see its diagram so that it can be easily understood by you. Now, just what we talked about, that 90% of the diet that we consume, 90% of fat that we consume in a diet is in the form of triglyceride. These are the triglycerides in the center, in the yellow three-limbed structure is known as the triglyceride. What you see around them, these green spherical balls, are actually the bile salts. So that means in the intestine, these fats are surrounded by the bile salts. Now what happens? As the bile salts surround them, these structures, lipases come into action, and these lipases come into action and start digesting these triglycerides, right? Hydrolyzing these triglycerides. Now, what about this structure? This structure in which triglyceride is in the center, bile salts are on the outer surface, along with the lipases, and along with these triglycerides in the center, it can also contain some fat soluble vitamins, the cholesterol and cholesterol esters. So this structure, this spherical ball like structure, is known as mixed micelle. Why we call it a mixed micelle? Because it can have variety of molecules. It is a combination of bile salts, triglycerides, phospholipids, fat soluble vitamins, cholesterol, lipase, etc., etc. So there are a number of molecules present within this one structure. So that is why we call it mixed micelle, and this mixed micelle is actually absorbed by the intestinal mucosal cell because its outer outpourings are hydrophilic, and in the center it contains some hydrophobic portions which are certainly lipid molecules. So these micelles are formed within the small intestine. They are disc-shaped clusters of amphipathic lipid. The important thing to remember is they have hydrophobic groups in the inside. These inside hydrophobic groups and hydrophilic on the outside. So this hydrophilic environment outside make them water loving so that they can be easily transported within the intestinal lumen and in the blood and reaches to their target sites. Okay. This is another representation of these mixed micelles. As you can see, some hydrophilic water-loving ends outside and hydrophobic insides. You can see cholesterol and triglyceride structures in the center, and this is the intestinal mucosal cell. And as you can see, these intestinal mucosal cells are readily absorbing these micelles into the enterocytes. So once the triglycerides or the fats are accumulated within the micelles, they are readily internalized or absorbed by the intestinal mucosal cells. Okay. Now we are in the small intestine, and just look how these micelles are actually formed. Okay. So in the lumen of the small intestine, what we consumed? We consumed triglyceride in our diet. Then we had some phospholipids, some cholesterol, and we can have some bile salts from the liver as well. And in the intestine, we have pancreatic lipase as well. As we talked about in the duodenum, pancreas releases some pancreatic lipases as well. So this triglyceride, which is absorbed or already there or synthesized within the intestinal lumen, these triglycerides are converted into two fatty acids, two monoacyl glycerol, or fatty acids, or cholesterol, whatever it is. Whatever the type of lipids or fats present in the intestinal lumen, what they do, they concentrate it, they gather somewhere in the intestinal lumen, and bile comes from the liver, which surrounds all these structures, and this bile salts, lipase and fatty acid structure give it a spherical shape, micelle-like shape. So this is how it is formed. It is synthesized in the intestinal lumen cell. What happens next when this micelle is formed? Okay, when this micelle is formed, it moves this way. It moves this way like a fan, like a wheel. It moves this way, and it moves to the intestinal brush border, and from the intestinal brush border, by the process of simple diffusion, it gets absorbed by the enterocyte, right? So what happens in the enterocyte? What happens in the enterocytes? Okay, what we talked about, that these micelles contain what? Either the triglycerides, the cholesterol themselves, phospholipids, and so many types of lipids. So all these are absorbed in the enterocytes, and what happens? Phospholipids, cholesterol, protein, all of the ingredients of the micelles are dispersed there in the intestinal enterocytes, and the fatty acids, which is coming from the triglyceride. This triglyceride is hydrolyzed or converted into 2 monoacyl glycerol and some free fatty acids within the intestinal enterocytes, and these are absorbed and released within the intestine. So what happens there? From the intestinal lumen, now they are into the intestinal cell. What happens there? From here, from the intestinal cells, these fats, whether in the form of triglyceride, whether in the form of cholesterol, whether in the form of phospholipids, whether in the form of fat-soluble vitamins, whatever the form of fat is, once they are there in the intestine, they need to absorb by the blood. And for the absorption, what do we have in the enterocytes? For the absorption purposes, we have chylomicron. We have chylomicron in the enterocytes, in the intestinal cells. And what is chylomicron? Chylomicron is a lipoprotein, is the first lipoprotein which is produced by the intestinal mucosal cell. And so these chylomicrons are absorbed, are taken up by the lymphatic system, which are known as lacteal, and they are taken up by the lymphatic system to the thoracic duct, and from the thoracic duct they are transported to their target sites, to their adipose, to the adipose tissues for their transport. So this is how lipids are absorbed. So the three processes you need to remember. One is the dietary lipid in the form of triglyceride that reaches to the intestine, where they are converted into simpler form with the action of bile salts and pancreatic lipases into the micelles. These micelles are water loving. Since they are water loving, they are easily absorbed by simple diffusion into the enterocyte. In the enterocyte, they release their content. They release their monoglyceride, their phospholipid, their cholesterol. These contents are taken up by the chylomicrons, and the chylomicrons are transport them into the lacteals or the lymphatic channels to the thoracic duct, and from thoracic duct to the blood, and then their target sites. Okay. This is the summary of this concept, if you can understand. Triglycerides, bile salts and lipases acts on the fatty acids, converted into monoglycerides. They are absorbed into the enterocytes, again reassemble, again make triglycerides. We will discuss it somewhere else in triglyceride synthesis. And these triglycerides again are taken up by the chylomicrons, and through chylomicrons, which are the lipoproteins, they are taken up by the lacteals and transported to the thoracic duct, and from there to the their target sites of the blood. Okay. What happens if there is some abnormalities of lipid digestion and absorption? Number one, cystic fibrosis. Cystic fibrosis is a genetic disease. It is an autosomal recessive genetic disease. And what happens in that, there is a mutation in the gene, CFTR gene, for the chloride channel on the epithelial surface. So as a result, what happens? There is decreased secretion of chloride and increased reabsorption of water. So as a result, the secretions from the mucous glands becomes thick. So as a result, secretions become thick. So this happens in the cystic fibrosis, and it can affect number of organs, bronchioles, trachea, pneumocytes, GI cells, pancreatic tissues, so that they offer some pancreatic enzyme disturbances as well, and replacement of these enzymes and fat-soluble vitamins digestion and absorption leads to abnormal digestion and absorption of fats by the intestine. And it also results in chronic lung infections if the respiratory system is involved. So if you can recall the case, the scenario, mother also complained of struggle breathing. At times the child was had difficulty in breathing, maybe due to the involvement of the respiratory system as well. So abnormalities of lipid digestion and absorption, yes, cystic fibrosis can also result in abnormalities of digestion and absorption. And number two, steatorrhea. This condition is known as steatorrhea. What happens in that? In that, there is increased excretion of lipids or essential fatty acids or fat soluble vitamins in the feces. And what is the cause? Cause is abnormal or disturbed digestion and absorption of the fats. Why abnormal or disturbed digestion and absorption? Because of insufficiency of pancreatic hormones, their lack or the mutation in CFTR gene, which leads to cystic fibrosis. Cystic fibrosis can present as steatorrhea, which is a lipid malabsorption disorder. Now, here it is shown that what happens in steatorrhea, the basic primary defect is the dietary lipids. When these dietary lipids reaches to the small intestine, so either there is lack of bile from the gallbladder. So if the bile is not there, so what will happen? There will be no micelle formation. Okay. Or either there will be lack of pancreatic lipase. So what happens if there is lack of pancreatic lipase? Two things. There will be no formation of the micelle, because micelles are formed by the fat droplets surrounded by the bile salts and lipases, and pancreatic lipase are the final lipases which offers digestion and hydrolysis of triglyceride present in the small intestine. So these defects can lead to steatorrhea, which is excess lipids in the feces. So these were the abnormalities of the digestion and absorption. If you have any questions, you can